Okay, thank you. Okay, and the name of the chapter is Cell and Organelles. Okay, what is cell, Fahad?
Cell is inside your body.
Well,
It's a unit, just a basic unit of life.
it's a basic unit of life. All the organisms in this world are made by cells, correct? Okay, now
Yeah.
these cells are not empty spaces. They are also made up of tiny structures named as organelles. Is it clear?
Yes.
It's their
Yes.
body
Oh,
parts, you know.
yes, their body parts, you could say. Yes. Okay, now here's the thing. The thing is a little bit history. Okay, Lamin, I love to ask you this question. Who was the person who created classification and naming system?
Yes.
Carl Linnaeus. Okay, now here is the thing. Who discovered cell? Any idea?
Not him, right?
Scientist.
Oh, thank you. I didn't know. Maybe he's a human. Okay, see here. Okay, the name of the person is Robert Hooke. Okay, the name, oh, I think the spelling got opposite way. It is Robert Hooke, not Ruhik Robert.
Rukobert.
Okay, it is Robert Hooke. Okay, I will fix it later. Robert Hooke is the name. In 16th century, this guy over here, can you see Robert Hooke? In 16th century, 1665, just remember 16th century, he was the first person who discovered cell. And what did he do? He was just sitting, chilling, okay, and he thought, why don't I see that this, okay, the trunk, this cork, he took a small piece of it and he put here, this was the microscope of 16th century, and he saw it and he saw this picture, okay? This, can you see over here?
Yes.
Okay, this picture. And he was amazed. He thought, wow, okay. Then he concluded that maybe all the organisms, okay, they are made up of cell. So the first term cell, Robert Hooke used it. Is it clear? Okay, do you know what's the meaning of cell? Amir? No?
Basic unit of life.
Well, that's a biological meaning, but cell, okay? Well, we call it, okay, take this thief to a cell, prisoner, something related to that. You got an idea?
Cell.
Yes, cell. So this person thought that these things look like a cell, okay? Look like a jail to him, okay? So he called them cell. So this was the person. This is the guy, guys. I am not the person who is teaching you. This is the person who created all these things. well discovered, okay? So Robert Hooke in 16th century, he made cell, not made, he discovered cell, okay? Now, the problem with scientists is that they will not stop. If they have given you one torture, they will try to give you more. Okay? So the same thing happens. Is my voice clear to all of you?
Yeah.
Okay, please show me thumbs up. Okay, great job. Now here, what happened then this scientist, can you all see? This scientist. His name is Anton van Leeuwenhoek. What's the name?
Anton van Leeuwenhoek.
What's the name, Fatima? Anton van Leeuwenhoek. What's the name, Fatima? Fatima?
Teacher is from France,
Anton
right?
van Leeuwenhoek.
Excellent. Amir, what's the name?
Anton van Leeuwenhoek.
Excellent. Fahad, what's the name?
Anton van Leeuwenhoek. I don't know. Okay, this
Yes,
stupid name.
Anton van Leeuwenhoek. Okay. Well, he's popular with the name of Leeuwenhoek. Okay, but his full name is this person in the same century, 16th century. Okay, what did he do? He improved the microscopes. He improved the magnification of microscopes. And okay, well, Robert Hooke just said that cells are empty compartments. Okay, is it clear? Okay,
Yes.
he just saw that cells, okay, they are not made up of any structure. They are just empty compartment named as cell. But this person, he just digged inside with new microscope, with new magnifications, and he said, bro, you are incorrect. Cells are not empty compartments. They might have something inside them. So this was the person who actually bring the idea of organelle. Is it clear?
Oh,
yes.
This guy was it.
Yes, this guy. So the torture begins. Second torture you guys have to face. Third one. So 16th century, Leeuwenhoek. Okay, so this was the picture. Let's suppose he just saw the empty compartment, but this person saw inside it. Okay, so this is one cell, and these are the structures inside it. Okay, now let's move
Yes.
on. Do you think torture is over?
Of course not.
Excellent job. Okay, here, two main scientists, Schledin and Sean. What's the name?
Schledin and Sean.
In 18th century, okay, well, in 16th century, 17th century, children were happy, okay, now we just have to study this. But in 18th century, Schledin and Sean came in and they said, bro, come on, do you know that all the plant cells are made up of cells and all animal cells are made up of cells? Okay, so in 18th century, 1830s, okay, two other scientists, what did they do? This one, Schledin, Matthias Schledin, he observed that plants were made up of cells, okay? Theodor Schwann, he observed that animals are also made up of cells. So this is their contribution. Is it clear? But the
Yes.
problem with this is, so I will ask you that what was the problem? What is the limitation? Okay, what was the thing which Schledin and Sean didn't discover correctly, you could say? They thought that, okay, plants and animal, both of them are made up of cell, but they thought that maybe cell are planted somewhere. So how do cell come into being? They thought maybe a magical alien will do it. Okay, so this was their concept, which was totally wrong. And with scientists prove them wrong, do you know? Any idea?
Carl Linnaeus. Carl
Oh, no,
Linnaeus.
no, Carl Linnaeus, no. Ask him any idea.
Nothing.
Lamin,
Yes.
any idea? Which scientist did it? Which scientist proved them wrong?
Of course, some stupid torturous scientist.
Okay.
Yeah. I think
Okay,
That's-
here, the name is Rudolph Virchow. Okay, the name is Rudolph Virchow. Here is handsome person.
I mean, actually looks good.
Yes, really? Lamin is like, okay. So 18th century, Rudolph Virchow, what did he said? He said, no, bro, you guys are wrong. The cell actually originate from the cells which are already existing. Okay, so this was his idea. Okay, so Virchow stated that every cell originates from another existing cell. Okay, such as all the humans come from the humans which are already existing. In the same way, cells will come from the cells which are already existing. Does it make sense?
Yes.
Well,
Yes.
the cell division and things we're gonna study in future. Okay, so how many scientists did we did? Okay, so cell theory and cell theory, cell history, or cell discoveries, these are the main scientists. The first one is Robert Hooke in 16th century. What did he do, Ayan?
Discovered cell.
Ayan, what did Robert Hooke do?
Discover cell.
Excellent, discovered cell. And when he was discovering cell, did he use animal or plant, Fahad?
Trees, oaks. Tree branches. I don't know what to call it.
Okay, we call it cork of tree, okay?
Okay.
Cork, C-O-R-K, cork of tree, okay? Amir, who was the next scientist who provided us with torture?
Van Leeuwenhoek.
Excellent, what did he do?
He improved microscopes.
He improved microscopes and what did he say? Cells are empty? No, they have some tiny creatures. Okay, Fatima,
Yeah.
who was the next two scientists?
I don't know the name, but they discovered that plant and animals are made up of cells.
Yes, and their name is Schledin and Sean. What's the name?
Schledin and Sean.
In 18th century. Okay, now Lamin, Virchow.
Every cells come from existing cells, right?
Okay, excellent. Now what happened? Their ideas, when we sum up, when we make 3 points of it, it give us a very important theory of biology named as cell theory. What's the name?
Cell theory.
Cell theory, okay? So over here, what is cell theory? Here, cell theory have 3 important points which these scientists have discovered, okay? And the first point is this: cell is the basic building block of life. Will you ever forget? This is the first point of the theory. Easy peasy, isn't it? Excellent job.
Yeah.
The second one, all organisms consist of one or more cells. Yes, Schledin and Sean already proved it, that animals and plants are made up of cells. So all organisms consist of one or more cells. Does it make sense?
Yes.
Yes.
Excellent. What's the third point? New cells may only arise from other cells or pre-existing cells. Does it make sense?
Yes.
So these are the 3 important points. Year 9, year 10, and 11. Remember, cell theory will be my main question in test. Okay? Any questions? Any questions so far? No? Sure?
No.
Sure? Fahad?
Yes.
Ayan? Fatima? Amir? So these were the scientists. Remember, the names are important. Century is important, okay? Here, cell theory, all organisms are made up of 1 or more cells. Cells are the basic unit of life. All cells come from existing cell, okay? You should know that which point is contributed to which scientist, okay? Most of the time people say that cell theory, okay, it was proposed by Rudolph Virchow and Sean and Schledin, okay?
Yes.
Well, yes, but remember, okay? Okay, next one. Next one I already taught you. It is one of the easiest topic. It is about microscopes. Yes, what is microscope? Come on.
Oh, you want an answer?
I'm expecting an answer.
Okay, okay. You can zoom into things which is not visible from a naked eye.
Oh, what a great answer. I didn't know. Thank you. Thank you. So
You asked it. What else do you want?
microscopes are of 2 types, okay? Light microscopes and electron microscopes. Lamin, what's the difference between light and electron microscope?
Light, they use light, right? To see
Oh,
the
yes, I know
stuff.
that. Okay.
In the electron microscope, they use electricity, right? To see through.
Well, yes, it's a good guess. Yes, they use electricity. I will accept it for now. Yes, in electron microscope, they do not use light, okay? Okay, but here.
Could have said it's more expensive.
Okay, so far you have studied that microscopes are of 2 types, light and electron. Fatima, is it clear?
Yes.
Mary, where are you? Okay, now here, electron microscopes, Asim, you can call your friends, the one who's going to do the presentation, okay, because this is the last thing I'm teaching you. Okay, electron microscopes are again of 2 types. Do you know the names? Any idea about them? Okay, the silence show no. Okay. The first one is scanning electron microscope, or the short form is SEM, and transmission electron microscope, the short form is TEM. Okay. Is it clear? Thumbs up from everyone?
Yes.
Excellent job. Okay, let's move on. Here, light microscope, I already told you in detail what are the things in that. Okay, quickly, Mary, okay? Okay, so light microscope, you already know this thing. Their parts. Well, this is the thing where you have to just memorize. Sorry, I have to say you. I know, well, I hate to memorize, but you guys have to. Okay, now the second thing is here. This is an electron microscope. Can you see here? Okay.
Yes.
Okay, electron microscope, this is a microscope with high magnification, okay? We can use this microscope to dig inside a cell, okay? There are many organelles which are not visible in light microscope, but they are visible in electron microscope. Is it clear?
Yeah.
Sure. No confusion, should I move on? Okay, I think it's pretty simple. Here is the thing, how do 2 microscopes work? Here, in electron microscope, okay, this is a simple picture. The source of the light is obviously a light, a bulb, a sunlight, any source. But over here, the source of light is electrons, okay? You will learn electrons in future, okay? Or maybe you have heard electrons in physics class. Have you? No? Electrons, protons, neutrons, no?
Yeah.
Okay, but electrons, okay? Over here, we have some objective lens. We do have an objective lens here, okay? Here, when our eye will be here, okay? But over here, our eye is here, not down. Over here, my specimen means the thing on which I'm doing an experiment, okay? The specimen is here, but over here in light microscope, the specimens are always in the middle, okay?
Yes.
So this is a basic difference you should know, okay? Just to give you an idea, okay? Well, the details, no, not needed. Okay, here's a comparison. The things which you could see with eye, with light microscope and electron microscope. Child, hand and finger, very easily visible with eye, isn't it? Or you need a microscope to see it. Let's suppose Lamin want to see Fahad's hand. He will bring a microscope.
You want to
Is
see my
his
hand,
hand
bro?
really tiny? Hey, yo.
Okay, but I don't think so. No. We just need an eye. Okay. Well, light microscope. If Lamin want to see a blood of Amir, okay, the detailed blood version, okay, then he have to bring light microscope in. But let's suppose if Lamin want to see DNA of Amir, he have to bring electron microscope to see that. Okay.
Yes.
Does it make sense? So these are the things
Yep.
which are visible, okay, or it was a comparison. Now the thing is, okay, Fatima, how many types of microscopes are there?
2 types.
Which one are they?
Light microscope and electron microscope.
Excellent. How many types of electron microscopes I have?
2,
2. What are the names?
Sim and Tim.
Wow, I remember, I knew that all of you will remember the shortest version, huh? It is scanning electron and transmission electron. What's the difference here? See this picture. Okay, well, this is not an impressive picture. Let me show you another one. Here, can you see? What is this?
Rich.
Wasn't it the same? Oh, okay. I just see this bug thing right now.
Yeah.
I think it's a fly. It's a fly.
It's a fly, it's a bug, correct? Remember, in
Yes.
scanning electron microscope, we use magnification, but it can only scan the pictures, okay? So if we want to see tiny creatures, we can just scan their body parts and we can create a picture such as this, the face of this bug, okay?
And why he looks angry? Poor guy.
Okay, now, but listen here, in transmission electron microscope, we can dig in, okay, and we can see the details of it. Can you see here? This is a detailed picture. Now, this is a pollen grain, okay. Hazik when he's coming, huh? Okay, pollen grain. This is the outer picture taken from scanning electron microscope. Scanning can only scan the outer surfaces, the outer boundaries, and give you a picture, but scanning cannot give you a detailed picture. Okay, transmission electron microscope. Okay, transmission electron microscope, it gives you the detailed picture. Okay, any questions so far? Okay, now all of you turn off your mics. Okay, now you will answer first. Okay, electron microscope, 2 types, scanning and transmission. Scanning only scan the outer surfaces of organism and give us pictures such as here, the picture of a bug. Okay. Transmission, it gives us a detailed version pictures where we can see, dig in what's present inside, okay? But the thing is that, okay, not these ones. Here are the differences of light and electron microscope, which you should know, okay? First of all, electron microscope, they can magnify up to 1,000 times, okay? Well, this one, light microscope, they only have magnification up to 1,500 times. That's it, okay? This is the important difference which you should know. This is the first one. Is it clear to all of you?
Yes.
Sure. No questions? So this was our class for today, okay? The thing which we learned, it was history, okay? History of cells. Secondly, we learned about microscopes. So let me ask all of you one by one. Okay, so Fahad, who was the first scientist who gave us this torture to study all of this?
Robert Hooke. Yes, Robert Hooke.
Robert Hooke. In which century?
1665.
Yes, excellent. Yes, 1665.
Bro,
16th
my
century.
man gave me the absolute answer.
Yes, impressive, huh? Fatima, who was the next scientist who did the work? Fatima?
Yes, teacher.
Yes, who was the next scientist who did the work?
Teacher, I forgot the name.
Okay, here is the name. Leeuwenhoek.
Leeuwenhoek.
In which century?
In 16.
Yes, 16th century. What was his work?
He discovered that cell also have tiny structure inside them.
Excellent job, Cell, and he modified microscope carefully. Amir, who were the next 2 scientists who worked after Leeuwenhoek?
Schledin and Sean.
Yeah, Schledin and Sean. In which century?
What?
In which century? In which century?
17th.
Oh,
No.
no. What was the century, Lamin?
18th, right?
18th century. Okay, and what was their work?
Schledin observed that plants are made of cells, and also that one Schwarm, I don't know how to pronounce his name, but he observed that animals were also made of cells.
Excellent job. Okay, Fahad, you. Who was the scientist? Next one.
What I did?
No, after
It was,
Schledin and Sean, who was the scientist who contributed?
Rudolph Virtue or something.
Excellent. Rudolph Virtue. In which century?
Yeah, I don't remember. I think 18.
18th century. Excellent. Okay, so cell theory have 3 important points. Remember, okay, cell theory have 3 important points. I'm sure that all of you know that. But over here, what's the difference between scanning and transmission electron microscope? Fatima.
Scanning electron microscope only scan the image of the organism, but transmission can also show you the detail.
Excellent
Picture.
job. Okay, so Fahad, can you tell me light microscope? Okay, what is the maximum magnification for light microscope?
1,000 to 1,500.
Excellent job, Fahad. Okay, Lamin, what is for electron microscope?
Electron microscope 1,000 to what's say
Okay, Amir.
I forgot.
Okay, it's okay. Amir. Here, it's written.
Was it 1,000 times?
Here.
Okay.
Okay. Yes, so this is the magnification. Okay, Amir, what's the difference between scanning and transmission electron microscope?
Transmission gives you more details.
And scanning give you. So remember, in IGCSE, they will put some pictures in front of you and they will ask you to write which is from scanning and which is from transmission, okay?
Yep.
Okay, so this thing, today's class is clear to most of you,